What is animal cell culture? Animal cell culture is the process of growing and maintaining animal cells outside their natural environment, in vitro, under controlled laboratory conditions. These cells are supplied with nutrients, growth factors, gases, O2 and CO2, and maintained at specific temperature, humidity, and pH to support survival, growth, and division. It is a fundamental technique in biotechnology used for studying cellular physiology, drug testing, vaccine production, cancer research, and recombinant protein expression. Cells can be cultured as adherent monolayers, attached to a surface, or as suspension cultures, floating in the medium. What are cell lines? Cell lines are cultures of animal cells that can be maintained and propagated for multiple generations. They originate from primary cultures and can be categorized as primary cell culture, directly obtained from animal tissues. Limited lifespan, finite number of divisions, usually 20 to 50. Retain characteristics of the original tissue. Secondary cell culture / finite cell lines. Derived from subculturing primary cells. Still have limited lifespan. Continuous cell lines. Immortalized cells that can divide indefinitely. Obtained through mutations or transformation, natural or induced. Examples: HeLa cells, human cervical cancer cells, CHO cells, Chinese hamster ovary, Vero cells, monkey kidney cells, BHK21 cells, baby hamster kidney. Importance of cell lines. Model systems for studying cell biology. Used in production of monoclonal antibodies, vaccines, and therapeutic proteins. Serve as test systems for cytotoxicity, carcinogenicity, and drug discovery. Equipment used in animal cell culture. For successful animal cell culture, a sterile and controlled environment is required. The main equipment includes A. Sterilization and aseptic equipment. Autoclave, sterilizes glassware, media, and instruments. Hot air oven, dry heat sterilization. Filtration units, membrane filters, sterilizes heat-sensitive solutions. UV lamps, sterilization inside laminar hoods. B. Aseptic work area. Laminar airflow hood, biosafety cabinet, provides sterile airflow and prevents contamination during handling. C. Culture maintenance. CO2 incubator, maintains 37 degrees Celsius, 5% CO2, and humidity for mammalian cell growth. Refrigerators and freezers, -20 degrees Celsius, -80 degrees Celsius, liquid nitrogen storage, for storing reagents, culture media, and preserving cell stocks. D. Analytical and monitoring equipment. Inverted microscope, to observe living cells in culture flasks. pH meter, to measure and adjust culture medium pH. Centrifuge, for separating cells from media or during washing. E. General lab equipment. Water bath, thawing frozen cells or warming media. Magnetic stirrers and shakers, for mixing media and suspension cultures. Glassware and plasticware, culture flasks, petri dishes, pipettes, centrifuge tubes. Components of animal cell culture media. A typical animal cell culture medium contains the following major components. 1. Inorganic salts, ions and buffers. Provide essential ions for cell survival, osmotic balance, and membrane potential. Maintain pH and regulate cell permeability. Common salts: NaCl, KCl, CaCl2, MgSO4, NaHCO3. Example: sodium bicarbonate, NaHCO3, acts as a buffer with CO2 in incubator to maintain pH 7.2, 7.4. 2 amino acids. Building blocks of proteins, essential for cell growth and proliferation. Cells cannot synthesize all amino acids, so medium must provide them. Example, L-glutamine, critical energy source, but unstable rightward arrow must be added fresh. Other amino acids, L-arginine, L-lysine, L-leucine, L-tryptophan. 3 vitamins. Act as coenzymes in metabolic reactions. Most animal cells cannot synthesize vitamins. Examples: B-complex vitamins, riboflavin, thiamine, folic acid, niacin. Ascorbic acid, vitamin C antioxidant role. 4 carbohydrates, energy source. Main energy supply for cultured cells. Glucose is the primary carbon source, usually 1 to 4.5 g/L. Some media contain galactose or other sugars. 5 lipids and fatty acids, optional. Some specialized media include lipids to support membrane synthesis, usually provided by serum. 6 serum, complex supplement. Fetal bovine serum, FBS, is most commonly used. Provides growth factors and hormones, attachment and spreading factors, fibronectin, laminin, binding proteins, transferrin for iron, albumin for lipids, detoxifying agents. Limitation: expensive, variable composition, possible contamination risk. 7 growth factors and hormones, serum-free media additives. For serum-free or defined media, growth factors are added directly. Examples: insulin, regulates glucose uptake. Transferrin, iron carrier. Epidermal growth factor, EGF, fibroblast growth factor, FGF, stimulate proliferation. 8 antibiotics and antimycotics. To prevent microbial contamination. Common ones: penicillin, streptomycin, against bacteria. Amphotericin B, against fungi. 9 pH indicators. Phenol red is commonly added. Red equals normal pH, 7.2 to 7.4. Yellow equals acidic, too much lactic acid. Purple equals alkaline. 10 water. Pure, deionized, sterile water is the main solvent. Must be endotoxin free. Examples of common media: MEM, minimum essential medium, contains amino acids, salts, glucose, vitamins. DMEM, Delbecco's modified Eagles medium, richer in amino acids and vitamins than MEM. RPMI 1640, widely used for suspension cultures, especially lymphocytes. HAMS F12, used for serum-free culture, includes extra nutrients. Steps in animal cell culture technique. 1. Preparation stage. Before culturing, everything must be sterile and ready. Sterilization of materials, glassware, plasticware, and media are sterilized by autoclave or filtration. Preparation of culture medium, appropriate medium, example DMEM, RPMI 1640, is prepared, supplemented with serum, like FBS, antibiotics, and L-glutamine. Work area sterilization, the laminar airflow hood, biosafety cabinet, is disinfected with 70% ethanol and UV sterilized. 2. Isolation of cells, source of culture. Cells for culture can come from primary cells, obtained directly from animal tissue, via biopsy or organ excision. Tissue is minced into small fragments. Enzymatic digestion, trypsin, collagenase, dispus, is used to separate cells. The cell suspension is filtered to remove clumps. Cell lines, thawed from frozen stocks or taken from existing cultures. 3. Seeding the cells. The prepared single-cell suspension is transferred into culture flasks, petri dishes, or multi-well plates. The vessels already contain pre-warmed sterile culture medium. The flask is gently swirled for even distribution. 4. Incubation. Cultures are placed in a CO2 incubator at 37 degrees Celsius, 95% humidity, and 5% CO2. The CO2 works with sodium bicarbonate in the medium to maintain pH. Cells adhere, if adherent type, and start dividing. 5. Observation and monitoring. Cultures are regularly observed under an inverted microscope. Monitoring includes cell attachment, morphology, shape, health, growth rate and confluency, percentage of surface covered, pH changes, indicated by color change in phenol red. 6. Subculturing, passaging. When cells become 80 to 90% confluent, covering most of the flask surface, they must be transferred to new flasks. Process. Old medium is removed. Cells are washed with PBS, phosphate buffered saline. Trypsin EDTA solution is added to detach adherent cells. Detached cells are resuspended in fresh medium. A portion of the suspension is transferred to new flasks. 7. Preservation, storage. Cells not immediately used are frozen for future use. Cryopreservation steps. Cells mixed with cryoprotectant, usually 10% DMSO. Frozen slowly, 1 degree C/min, to 80 degree C. Transferred to liquid nitrogen tanks, 196 degree C, for long-term storage. 8. Quality control. Ensure absence of contamination, bacteria, fungi, mycoplasma. Confirm identity of the cell line, DNA fingerprinting, karyotyping. Applications and importance of animal cell culture. Animal cell culture is one of the most important techniques in biotechnology, medicine, and life sciences. Its applications range from basic research to large-scale industrial production. 1. Basic research applications. Cell biology studies, understanding cell division, signaling, differentiation, apoptosis, cell death, and gene expression. Cancer research, studying tumor cell growth, oncogenes, and drug resistance in cancer therapy. Virology, virus isolation, replication studies, and development of antiviral drugs, since many viruses require living cells to grow. 2. Medical applications. Vaccine production. Polio vaccine, rabies vaccine, hepatitis B, and influenza vaccines are produced using cultured cell lines, example Vero cells, CHO cells. Monoclonal antibody production. Hybridoma technology uses animal cells, B lymphocytes fused with myeloma cells, to produce highly specific monoclonal antibodies. Recombinant protein production. Therapeutic proteins such as insulin, erythropoietin, interferons, clotting factors are produced using mammalian cells. CHO cells are widely used. Gene therapy and regenerative medicine. Cultured cells are used as vectors for introducing therapeutic genes. Stem cell cultures help in regenerative medicine, repair of damaged tissues, bone marrow transplantation. 3. Pharmaceutical and drug development. Drug screening and toxicity testing. New drugs are tested on cultured animal cells before clinical trials. Reduces the need for animal testing and provides reproducible results. Personalized medicine. Patient-derived cells can be cultured to test drug responses individually. For biotechnology and industrial applications. Production of biopharmaceuticals. Large-scale bioreactors with cultured animal cells are used to produce proteins, enzymes, and hormones. Hybridoma technology. Used in diagnostic kits, ELISA, pregnancy tests, infectious disease detection. 5. Tissue engineering and regenerative medicine. Artificial organs and tissue repair. Cells are cultured on scaffolds to form tissues such as skin grafts, cartilage, or even experimental artificial organs. Stem cell research. Culturing embryonic or adult stem cells helps in understanding development and in developing therapies. 6. Genetic engineering. Transfection studies, introducing foreign DNA/RNA into animal cells to study gene expression. CRISPR/Cas9 research, cell lines provide model systems for gene editing experiments. Recombinant vaccine development, example, hepatitis B recombinant vaccine. 7. Toxicology and environmental testing. Used for testing cytotoxicity of chemicals, cosmetics, and pollutants without involving live animals. Regulatory agencies often recommend in vitro cell assays before animal/human trials. Importance of animal cell culture. Provides a controlled, reproducible, and ethical alternative to using whole animals. Plays a central role in biotechnology, medicine, and pharmacology. Enables study of diseases, like cancer, viral infections, in a simplified environment. Essential for the production of modern therapeutics and vaccines. Supports advancement of regenerative medicine and personalized therapies.